Welcome to the Lessons from Lab and Life podcast. I'm your host, Lydia Morrison, and I hope that this podcast offers you some new perspective. This is the first episode in our COVID-19 Researcher Spotlight series, and today I'm joined by Brian Rabe, who's currently a graduate student at Harvard Medical School, where he normally studies retinal development. As the coronavirus spread, Brian decided to take some of the new technologies he'd been using to explore gene regulatory networks, studying tiny little embryonic mouse eyes, and apply them to improving and advancing coronavirus detection methods. Hi, Brian. Thanks so much for joining me today.
Thanks for having me.
So I was wondering if you could tell us a little bit about your recent medRxiv publication.
Yeah, so it's kind of funny. I usually study actually retinal development, but given the current pandemic, and I'd heard about this RT lamp technology from NEB, I decided I would try my hand at it before we closed everything down, and I got some promising results initially. So I actually got to stay on and pursue it. It's nice because it's essentially an alternative diagnostic assay for the virus. That's proven to be quite sensitive, and it's really, really fast. And what's nice is you don't need any specialized equipment to run it. So unlike a lot of the QRT PCR tests that are available that need really expensive machines and a lot of personnel training to run, this is an assay that could be run, potentially in a van with a heat block. So we really wanted to increase accessibility of testing, and I think that's what we've been able to achieve.
Yeah, so how were you able to achieve that?
Yeah, so it began just really just ordering these RT lamp reagents from you guys from NEB, and then designing several different primer sets. So designing them to hopefully work. It can be a little hard to predict which assays will work and which ones won't. And we got lucky, and actually the first one I designed worked really robustly. It worked very quickly, so the amplification happens very, very rapidly, and it was a clear readout. And importantly, it had a very low background. So a positive result with this set of primers does appear to be, pretty much 100% of the time a real positive, which is definitely necessary if we want to move into the diagnostic field.
Yeah, that's fantastic. So how quick is quick?
So from actually adding your sample to the reaction, it only takes 30 minutes. And then you get a very clear readout. I think, sort of peeking at reactions as they run, a lot of them will turn even sooner. But for maximum sensitivity, we found 30 minutes works really well.
Oh, that's great. And what kind of samples are you working with?
Well, for my work, it was all reconstituted samples. So I work in a biology lab, in an academic lab, and especially as this whole pandemic was progressing, there was no way I was going to be getting direct access to patient samples. I'm not medically trained. I'm not at a hospital. So what I was working with was an RNA control that came from actually Twist Bioscience, and then I reconstituted samples. That's how it basically means I stuck a swab down my nose many, many times to make essentially samples, whether they were nasopharyngeal or oral swabs or just collecting saliva in a tube. And then I would spike in these control RNAs to sort of serve as a fake virus in these samples to see would the reaction still run with things like mucus from a nasal swab or saliva, to see how well they would run and see how low I could push the amount of RNA that I was adding and still get a result.
And you mentioned the ease of the readout and the color change. Could you tell me a little bit more about what the readout in this LAMP reaction is?
Of course. So one of the really nice things about LAMP is that it generates enormous quantities of DNA. And the quantity generated is so enormous that every single base being added kicks off a proton, and with so much DNA being added, or being polymerized, you end up actually resulting in a pH change as long as your solution is buffered. So the way this whole system works, and you can read about this more on the NEB site, is it's a non-buffered reaction with a pH indicator dye that turns from red to yellow when the solution is acidified. And it makes the readout really, really clear. I mean, it goes from being this sort of purplish red to being a bright yellow. And the nice thing with this primer set is it's really been very binary. There have been some reaction or primer sets in the past that people have been trying that they sometimes get these sort of orange reactions that are kind of right on the line. But the nice
Sure.
thing about this primer set is that it's been really, really binary.
Well, that's excellent. And how did you get involved in this project?
Oh, well, a lot of credit there goes to my principal investigator, Connie Sepko. Basically, I knew about this reaction mix that you guys have been making for a while. I'm kind of, I'm a molecular biology nerd, so I read through catalogs from all these companies, and I like to keep up to date with what's going on. And I remember seeing that and thinking, oh, that's really cool. I'd love to try that sometime, even though it has nothing to do with what I normally work with. But then, this whole pandemic hit, and I realized that that maybe we were a little behind in developing new tests. So I just went to my boss and I said, I have this idea. Do you think I could try it? And being the supportive mentor that she is, she said, go for it.
Well, that's awesome. And how are you addressing sensitivity with the assay?
Yeah. So what's nice is that the primer set that we developed appears to be really quite sensitive, at least when I'm using RNA controls. The trick when we went over to clinical samples with our clinical collaborators over at MGH is a protocol, sort of a sample preparation step that comes before the assay, a really rapid step based on a protocol that actually came out of the Broad called Hudson that does a couple of really neat things all in one step. It inactivates all of the virions in your sample. You're basically boiling your sample with a few chemical additives. And that's really nice because it makes the sample safe for the people who are actually running all the tests. At the same time, it also breaks open all of these viruses so that they actually release the genomic RNA, which is what the sample is, or what the reaction is actually testing. So it makes the RNA accessible to the enzymes. And then it also inactivates patient enzymes, samples, things like RNases, that would immediately destroy that RNA under normal circumstances. This protocol also completely inactivates them, and we found that the sample is actually quite stable after that. And so the nice thing there is that alone, combined with the primer set we've been working with, has given us really impressive sensitivity down to, say, 40 virions per microliter. And I think
Wow.
it's been really impressive. We've been really grateful for our clinical collaborators' help in determining that. And what's I think is sort of next in the research pipeline, although not so much what I'll be doing, but what a lot of people are looking at now, is what level of sensitivity do you actually need in order to identify people who are infectious. And I think there's growing data that, of course, out of an abundance of caution, we've been anyone who throws any sort of positive, with the QRT-PCR tests, which are even more sensitive, that we're considering them to be infectious, but we don't really know. On the other hand, we also developed a really crude purification and concentration protocol that uses some really widely available reagents, things like basically glass powder that's used in the ceramics industry. And a nifty little protocol that's really, really cheap and really easy to run, and in most cases doesn't even need a centrifuge, and can allow someone to further concentrate the RNAs found in a sample to the point that you can increase that sensitivity down to even 1 virion per microliter. At least that's what it looks like with the RNA controls. And what's nice about this and sort of why we were able to do it is the LAMP reaction has proven itself to be very, very robust to a lot of things that would normally inhibit enzymatic reactions. And so we're able to do this really fast and cheap and crude purification, and the reaction is still able to tolerate any small amounts of inhibitors that get carried through, and that's been really impressive.
Yeah, I was going to ask about that. So the additions of chemical agents to help lyse the cells and prevent RNA degradation, those don't have any effect on the color change or the buffers or the other components of the reaction?
No, so you do. I had to fiddle around with them to make sure that the reagents I was using were at the proper pH when they're added. But luckily, they don't appear to have enough buffering capacity or anything like that to throw off the reaction.
Well, that's incredible. So what kind of samples have you tested so far? I know your paper was mostly focused on nasopharyngeal swabs, either sort of with or without this RNase protection treatment. But have you tried other samples, or is that something that's coming in the future?
Yeah, so I've actually tried, I think this is something as far as clinical samples that's in the pipeline, but I've tried with reconstituted samples straight saliva. And that's been
Wow.
something else where LAMP has I think, outperformed certain other tests in that the saliva doesn't appear to be particularly inhibitory to this reaction, especially when you run this inactivation step first. I think a lot of other reaction types that are sort of assays that are being developed tend to be much more sensitive to those, but the two enzymes in LAMP appear to be incredibly robust.
Wow, that's amazing and really promising to hear. So, where do you see COVID related research heading in the near future?
That's a good question, and I've been talking to a lot of people who are involved in this. Well, on the one hand, we're sort of creating. There's a group led by Mike Springer at HMS that's going to be comparing a lot of these assays head to head. So there will be several
Mm.
different LAMP assays. So the primer set that I developed, as well as some that have been developed at NED and Cornell, and I believe at least one or two other places. Then Mike Springer's lab has come up with an RT-RPA based assay, and then there's a CRISPR based assay coming out of the Broad, although those either have LAMP or RT-RPA back ends. And so they're going to be testing those head to head with the exact same clinical samples in order to get a really good idea as to how they compare. Outside of the actual diagnostics, there's also been a lot more work and more attention being paid to sort of upstream. Everything from, can we get enough swabs? So do we need to go to saliva, or can we stick with swabs, to what sample types actually have enough virus in them to detect well? Can you swab other places? Can you just have someone spit in a tube? And also sort of perfecting a lot of the collection protocols. We've been talking a lot about, what can a patient do themselves so that they reduce the risk of infecting whoever is testing them. Obviously we think a patient can spit in a tube on their own and probably close it and spritz it with disinfectant. So we're looking at things like that, and this is sort of leaving my wheelhouse very, very quickly. But there are a lot of people who are focused on this. And then downstream of this, people are looking at ways of sort of connecting these results to contact tracers and things like that, so that we can hopefully contain any potential sort of little outbreaks that may occur in the future. And then there's just, how do we deploy these things? Do we collect at a lot of different places and send samples to one centralized location for testing? Do we send sort of little pop-up labs to a bunch of different places? Things like, you were worried about a school, could you send a team in a van with heat blocks to a school and have them run tests? How do you do surveillance, things like that? So there's a lot of thought being put into that as well.
Oh, that's really interesting. Yeah, I often wonder, when we're going to be at the point where there's sort of a home test that someone could do. Someone could, spit in a tube at home, and could that tube potentially contain the right reagents, or, maybe you need to aliquot some specific volume. Do you envision this being something that eventually could move into home use, or maybe you spit in a tube and put it in the mail?
Yeah, I think any of those are good options. If you have a sous vide at home, it's very easy. We've actually tried that. Just a immersion heater in water, and you set it to 65, and that's really all you need. So yeah, I think a lot of people have been looking at how you would sort of formulate a kit, or maybe a device to make it easy to run. But yeah, I think a lot of people are looking at home diagnostics.
Yeah, and I'll be really interested to see that study from the Springer Lab of HMS come out, because certainly there's been a lot of fast and furious movement in terms of developing diagnostics to detect COVID. So it'd be really great to sort of see those compared in terms of specificity and sensitivity.
Oh, absolutely. And I have to give a lot of credit to Mike. He's put together a great group, and he's really, really built a sense of camaraderie. The way he sees it, his test may work in a different way than ours, but he likes to think of us not in competition, but as sort of supplemental to one another. We have assays that work in different ways. They may have different pros and different cons, and really importantly, different supply lines. So potentially, if we need to be producing millions of tests tens of millions, even hundreds of millions a week if you're thinking worldwide, having a number of different tests that are being made in different ways could be really helpful.
Yeah, certainly. And I think that that's a great example of the global camaraderie that's really come out of developing diagnostic tests and the free sharing of scientific knowledge, that's really surrounded the crisis.
Definitely.
So, Brian, did you find that compared to your normal research, there was a particular sense of urgency with this project?
Oh, absolutely. I mean, definitely, we were always thinking that this pandemic is progressing. Every time you check the news, you see that, it's spreading, and yet our testing capacity is not growing enough to meet the needs. And then, of course, states are beginning their reopening, and we're worried that without enough testing capabilities, we could just be looking at another outbreak if we don't catch things quickly. So there was definitely a lot of urgency. What also I think helped was working with other groups developing these assays. We were all motivating one another. We were sharing results. It became a really fluid and very rapid process.
So tell us something good. Do you have any good stories or reflections about your time in the Massachusetts sort of stay-at-home recommendation that you could share with our audience?
Well, I can say that it's been nice to see it in a lot of places. Like I'm out here in Waltham, and people are definitely taking things very seriously. I do see people are really practicing social distancing. I know you can hear on the news about things happening at various grocery stores and all that, but here it's been I think everyone's been sort of we're in this together kind of spirit. Those sort of drive-by graduation or birthday party celebrations have really taken off here. I think every evening we see 3 or 4 of them go by. So it's nice to see people supporting one another in a safe way. And beyond that, I mean, it's been nice to keep all the plants in my lab alive.
Lots of time to sort of focus on some of the more simple things in life.
Oh, absolutely.
Well, thank you so much for joining us today, and thank you so much for your work. I think it's been really important that you picked up this project. You were obviously able to identify some primers that made a huge difference in the sensitivity and specificity of the test. And I think that it'll go a long way towards really bringing a COVID diagnostic to more places, to more rural places, that might not be able to afford the instrumentation to carry out some of the RT-qPCR tests that are more prevalent now.
Oh, definitely. And having a lot of family back in rural Kentucky, that's definitely been something I've been thinking about.
Yeah, it's nice to sort of know. It's nice to know where people's motivations lie, and I think a lot of our motivations lie in protecting our friends and family and loved ones from this pandemic. So thank you so much for your work toward that.
Oh, well, thanks for having me.
Thanks for tuning in for this episode. As always, check out the transcript of this podcast for helpful links to further resources. And please join us for next episode when I'll be interviewing Feng Zhang of the Broad Institute, along with Jonathan Gutenberg and Omar Aboudier of the McGovern Institute of MIT, about their recent publication describing Stop COVID, an assay which combines isothermal amplification with CRISPR-based detection methods. So be sure to catch the next episode and hear from the scientists currently making major breakthroughs in coronavirus detection and diagnostics.